in a safer neighborhood. So I spent most of my years in Riverside, and as far as my family goes, I'm one of 5 kids. I'm the middle child. I have 2 brothers and 2 sisters. And my mom and dad are from Mexico. They're from Guadalajara, Jalisco. And my dad mostly worked in construction, and my mom mostly stayed at home. Sometimes she would take odd jobs here and there, but mostly my dad liked her to stay at home. So, and then I went to John W. North High School, and I was class of 2004. And so this was mostly in the school statistics at the time that I did this research was it was 18% Caucasian, 61% Latino, so it was mostly a Latino community, 13% Black, 5% Asian, and 3% other. And so North is, despite it being predominantly Latino and not necessarily like an affluent neighborhood, it is one of the few schools in California that awards this IB diploma, so International Baccalaureate Diploma. I don't know if you guys have heard of it. It's sort of like the AP program, but it's international. And this is awarded to like less than 10% of the students that are at this high school. And really my theory is because so John W. North is really close to UC Riverside. And so a lot of the university professors, you know, like they want good programs for their kids. And so this is one of the programs that really is unfortunately was implemented mostly to benefit the children of the university professors. Right? So it was unfortunate that it wasn't like advertised more broadly, because obviously the ratio of the IB diploma recipients was not 61% Latino, 13% Black, 5% Asian. Right? But somehow my word reached to me, and I applied to be in this program, and I graduated from North with the IB diploma. I went on to UC Berkeley, so I'm putting the years there so you guys can start counting up how long I've been in school. It's been a very, very long time. I was 4.5 years at Berkeley doing my bachelor's in mechanical engineering. And then another 5 years doing a PhD at Caltech, and I graduated there in 2015. And then I wanted to go into academia, into a faculty position, so I had to do this thing called a postdoc, which is these studies that you do after you get your PhD, and you do it for like very low pay. But you have a PhD, but anyways, it's almost like just exploitation. But 1.5 years in biosciences. So you can see I was like, my degrees are in mechanical engineering, but I worked in a wet lab because I wanted to learn biology. I was like interested in biology. I am interested in biology and synthetic biology. So I went to this biosciences department and learned how to like clone and do all kinds of fun stuff in the lab. So I was there 1.5 years, but that was in Houston. And I really, really wanted to come back to California for many reasons. And so I got a second postdoc at UC Berkeley, and that was in electrical engineering and computer science, and then I was there for 2 years. So if you add them all up, it's like, what is that, 4, 10, 11, 12, 13 years. 13 years to achieve my dream career, job, well, whatever, you know, where I am now as assistant professor of applied mathematics and statistics at UC Santa Cruz, and I've been there 1 year. So it's a very, very long career trajectory. And I guess one of the secrets to being able to persist for that long is that I just always really loved being in school. I've always resisted getting a job because I don't like having a boss, and I don't like having a schedule, and I don't like people telling me what to do. And so really like, it's been quite a journey, but it's been a lot of fun for me. And it has suited my personality type, and mainly in avoiding work. And and so now it's a little bit more work now that I'm doing the professorship, because you have some accountability, because people are going to care if you don't show up to class and lecture, right? And up until then, really, it was all the accountability was on me. Right? Nobody was holding me accountable for anything. But so what did I spend 5 years in grad school doing? My friends definitely thought I was some sort of genius. So she's on Caltech. I don't know if you guys watch The Big Bang Theory or not. That's Caltech. My mom, of course, like she'll call any time of the day that she calls me. She'll be like, the first thing she'll call me, and I'll answer the phone. She's like, "Oh my God, I'm sorry. Are you in a meeting? Are you studying?" I'm just like, I went to pick up if I was in a meeting, but no, it's okay, Mom. I can talk. So she thinks that I'm just like studying 24/7, and is always worried that she's bothering me. And then how society sees me. Yeah, you know, like there's that perception of when you go to grad school, you're like, you know, you're just kind of goofing off, right? And there are a lot of social hours, I'm not gonna lie, but we need it to keep our sanity because we cannot just be working 24/7, right? So you need a little fun time. And how my advisor sees us, I'm guessing there's like a bunch of lemmings over there. Yeah, we're one in a million to them sometimes. It feels that way. I don't know if that's how I see myself, but when I actually do cry at my computer, that happens a lot. Maybe not literally crying every now and then, but I mean, it's like emotionally a very taxing experience, but very fulfilling. And I was lucky enough I shared a room or an office in graduate school with a couple of other graduate students who happened to be female. So it was fun. There's like 3 of us in a room and 1 male. And we would spend a lot of time just griping, you know, just letting it out. And like, oh, like today was the worst day, this is what happened, and I'm worried about this, I'm worried about that, and just listening to each other. And so really like having that support group was essential also to getting me through grad school. And so, aside from wanting to quit all the time, which does happen, and maybe last year I wanted to quit my job too. It was just, you know, every time you go into something that's hard, you feel like you want to quit, right? I mostly learned a lot about controls, and this is the study of getting systems to do what you want them to do. And the applications are a lot. So, most of you guys probably are not aware of what you're probably not aware of how control theory influences your life and everyday life, right? So one major, huge example is flight control, right? When you get on a plane, you probably take it for granted. You probably just imagine that the turbines kick in, starts pushing air one way, and so that makes the plane go forward, and then it lifts up, and that's it, right? And then you just kind of steer it one way or the other. But if you think about it, there's a lot of things in the environment when you're on a plane that you cannot control, right? It could be something like a bird flies into the turbine, and one of the turbines gets stuck, and then you only have another, you have a backup turbine, so it's not the worst thing that could happen. Or you can have like gusts of winds, when you have turbulence, all kinds of environmental conditions that you cannot control, right? And so what's going to fix that? So back in the day, you might have just relied on a pilot, but pilots are now known to, well, they have a reputation for not having to do much now, because we have all these really fancy algorithms that will automatically adjust the plane actuators in order to make sure that the plane is stabilized and doesn't go fall crashing into the ground. And so I have a pointer. So they're very subtle actuators that you can't see, but there's a couple, I don't know if you guys ever paid attention when you take off, there's a couple on the wings here, flaps that will go up and down. And there's a couple rudders here in the back too. They're very small, you wouldn't notice them if you didn't pay attention, but their slight movements are very critical to keeping the plane on path. And so these algorithms will have sensors that figure out, well, this is the way I want the plane to go, but these external disturbances are pushing me this other way. How do I shift my actuators, or how do I shift the actuators so that I can stay on track? Another one is weird natural examples of control systems, because the things you need for control really is you need sensors, because you need some way to take in information from the outside world. You need the control system, in this case your brain, that's processing that information. And then you need an actuator, which then is like, I process this information, I'm now I know what I need to do next. How do I actuate that next action? And so that could be you turning the wheel if you see an obstacle, or it could be you stepping on the brakes, right? And nowadays we're like, oh, everybody's talking about these self-driving cars, right? And all these self-driving cars are trying to do is they're trying to replace you as a human and trying to replace your remarkable capabilities of being able to sense and react to the world outside and around you. And like really, all this machine learning, AI, cruise control, all those things are all us just trying to replicate how amazing humans are, right, into a machine. So I'm going to start off with a little bit about my PhD trajectory. So as I mentioned, a lot of my focus was on just understanding these concepts of control theory. What is control theory? What is the math that we use in analysis and design of these systems? But then you got to go a little bit deeper than that, right? It's not enough for me like to just have that knowledge. That's enough for me to teach some undergrad, graduate level class on the topic. Then you got to zero in on a specific application. Like what new application are you going to bring to the table using this new knowledge that you gained? And then, but most of the time, you're like, up until here, and this is time as you go forward, and this is kind of like the focus of your work from this broad top to the bottom. Up until where it says human knowledge, like this, all above is things that, information and knowledge that already exists in the world. But you're just trying to gather that knowledge that is pertinent to the problem that you're trying to solve. And then once you make finally you make a little dent, I don't know if you can discriminate, but a little dent past where it says human knowledge to PhD thesis topic, that's your dissertation, and that's your thesis, and that's where you say, this is the knowledge that I contributed to the world, right? This is a piece of knowledge that did not exist before I entered into my PhD program and did all this work and all this research and created this one piece of knowledge to share with everybody. So it's really, it's building the foundation, the basic principles, everything you need just to generate a new piece of knowledge. But it's pretty incredible to think that when you do something, you can say that no one in the world has done it before, as far as you know. Right? There has been cases where things have been done before, but people didn't know. Like hundreds of years will pass, and they'll fish up a paper from like the 1900s, but it's like written in Russian, so nobody knew that this paper existed, where they did this thing that you just did. But aside from that, it's not big deal. So what is a PhD in general? Like it may seem daunting, you might think that you have to have like a certain amount of ability or IQ, but it really is just, if you break it down into its parts, anybody could do a PhD. It's just a matter of if it's something you want to invest time in and if it's something that you enjoy. So it's just like a really long science fair project. Right? The science fair project takes you 5 years to complete. But the method is very similar to when you think about constructing a science fair project. It's just more involved and it takes a lot more study. And like I said, you build this foundational knowledge. So what is the broader topic of your interest? And find a gap in the knowledge that you would like to fill. And depending on how critical that gap is, so if it's a very critical gap, as in if I fill this small gap, this is going to accelerate this field of science 100 times, then that's like going to be an important, that's going to calibrate how important or how impactful your research is to the field. Right? And it doesn't, and it won't, coming out of their PhD, it's very likely that you won't have that huge of an impact, but that's okay, that's normal. Very few people will have a thesis that has a huge impact in science, but that comes later with more practice and more knowledge. And actually your ability to be a really good scientist is mostly founded on how good you are at figuring out what are the critical gaps in knowledge and asking the right questions. Asking the right questions is what really makes you a star scientist and a star researcher. And you become an expert in that gap. So then you're the go-to person. And it's not because you're so much smarter than the next person and because the other person didn't do it. No, it's because you invested that time to learn the knowledge that you need for that, for that gap of information, right? So you're just invested your life in becoming this world expert in this one area of science. And you create new knowledge, and you have to share it with the world. So that's what we create knowledge for. So, as I said, what does success in graduate school look like? You have this very deep knowledge of this very specific topic, and only a small community of people are really going to be able to understand what you're saying and assess whether you're correct or incorrect. So it starts to get fuzzy as you get, I know as I've gotten more senior, and now where I'm like the senior author on research papers, it's a little nerve-wracking because it's harder and harder to find people to fact-check you, right? There's no solution manual out there. No one, I have to be held accountable for my own stuff and check my work and make sure that it's correct. And so in general, industry does not care about the very specific knowledge that you hold, but they do care about the skills you acquired in the process of obtaining a PhD. So you shouldn't be so focused about what exactly your PhD studies are, but more in gaining the skills in the process. And they do appreciate that, because one of the things you learn is you learn to formulate and answer research questions. And that's a hard skill to develop. You develop communication skills, right? And then nowadays it's becoming really important because it used to be that people valued other valued workers with like high IQs because they could retain a lot of information, they could recall things at the drop of a dime. But if you think about it, now we have the internet. We have access to everything online, right? We don't need people to be able to memorize everything in a math book or every derivation, right? We need somebody that can see the big picture. We need somebody that can bring ideas together. We need somebody that can communicate those ideas, who can build a vision. So really this advancement in technology is changing the needs in industry and research. And also I developed tough skin and resilience. I'm sure most of you, I don't know if you haven't, there's like this really famous TED Talk that keeps going around about grit, right? And how important grit is in being successful, and that this seems to be the #1 indicator of success. And honestly, like I learned that with my friends going through grad school, and now it's been like, I guess, scientifically shown that this is in fact what leads to success. And I guess like coming from my background, that's easy enough to get. So my trajectory after grad school. So I wanted to broaden the scope of my work then. My application of works in control of dynamic systems, so I wanted to apply to genetic networks. So I got very interested in biology, thinking about controls in biology. So what has been done in the field of controlling genetic networks? And I'm going to introduce this topic a little bit because some of you may or may not know what it's about. So synthetic biology is just like, it's a relatively new field, depending on how you want to define it. But it's creating biological networks from the ground up. So you can take genes that already exist from different organisms, and you can put them together so that they can interact in ways that do not exist in nature. So for example, this is one of the first, what did I do? I pressed the stop button on accident. This is one of the first dynamical systems that was built with genes. It's an oscillator. And so it's a 3-gene oscillator. And so these genes, they express these proteins. And so what you see here is a measurement of the protein concentrations inside the cell. And so A is a protein that represses the production of this protein C, and C represses the production of protein B, and then B represses the production of protein A. So it's called a repressilator because they repress each other in a circle. And this has this like oscillatory dynamic. So you see they kind of go up and down, up and down. And so this was one of the very first demonstrations of building a genetic network from the ground up. And this was in the year 2000. And they just took these 3 genes, they cloned them, they assembled them together, and they put them in a bacterial cell with a fluorescent reporter. So I don't know if you guys know what fluorescent reporters are. They're these proteins that came from jellyfish, actually. So that's what the thing that makes jellyfish glow. That's what like really transformed single cell analysis in biology, because using these as a reporter, we can now see concentrations of proteins going up and down because they're glowing this green fluorescence. And then on this other end, another circuit that was built at the same time as the oscillator that was also famous is this toggle switch. So A represses B and B represses A. And so it's called a toggle switch because it can only be in one of 2 states. Like either B is, there's a lot of protein B, and since it's like pushing down A is at 0. But then if you switch it over to the other way, then A can be really high, and then B will be really low. So that's what's called toggle switch. You could switch it one way or another, where one is high and the other is low, and vice versa. So, I mean, it seems kind of trivial now if you think of the way I explained it, but it's actually quite remarkable. And so what they did more recently, this circuits were built in the year 2000, and that was back when people were like, yeah, this is cool, it's like Legos, but what use is that to humans? Like, what are we going to use synthetic biology for? And it was very unclear that synthetic biology had any real applications in the real world, right? But in 2014, they were like, well, why don't we use it to control, for say, example, therapeutics? Like, if you have a therapeutic protein that can help you suppress some kind of immune disorder or disease, right? So they put the toggle switch, this like synthetically, constructed man-made circuit, into the gut of a mouse, and then they were able to control whether B was expressed or A was expressed just by feeding the mouse sugar. And so that's like the bottom 2 curves. The yellow curve is with no sugar, and then the blue curve, you can see it goes up when they add sugar. So they induce the expression of one of the proteins. And so then the idea is that like, oh, we can put this in the human gut, and then we can control when they produce or don't produce a certain therapeutic protein. This is like kind of one of the first implications of a possible medical application of synthetic biology. Another thing that synthetic biology is big on, and this is really kind of big in the Silicon Valley area in the biotech industry, is enzymatic pathways, and that's how they do drug production or like biofuel production. And so this is just the top little Pac-Man looking things are enzymes, and they catalyze these reactions, right? So then you start with some substrate, and usually it's a sugar, and these different enzymes will take that substrate and will turn it into something else, and the next enzyme turns it into another structure, and finally you get your final product out. And so a lot of the research that goes on in the biotech industry is trying to figure out how to get the most of the final product with the least amount of sugar possible, because you want to reduce your expenses and get a really high yield. Like at Stanford, they built a pathway that created opioids. So they can, like, in the lab, they'll just generate opioids just by these synthetically constructed enzyme pathways that they made. JBay does biofuels. One of my friends works at Amyris, and they created something that's like a moisturizer, and now they sold it to like Sephora, so they're getting really rich off that now. So this is like one of the bigger ones that are definitely making money now. So synthetic biology has a potential for a really high impact across many fields. And the idea is like if we can control biology, because one of the biggest problems with biology is that it's hard to control because we don't understand it, and it does all these crazy things, and we try and get it to do one thing, but it does something else. So even like those small circuits that I showed you, at this point, it's really hard, without knowing more background information, it's hard to appreciate how hard it is to build that. But if you can get it to do what you want to, it's like the applications are limitless. You can do smart bandages, meaning that we can get like wounds to heal faster, limbs to regenerate, we can grow hearts, right? We can do all these things. If we can control how cells behave and differentiate and all these things, we can like grow a human being, right? Another thing is meat culturing. One of my former alums, she did her master's with me. She now works at Memphis Meats, and they culture meat so it's supposed to be an environmentally friendly way of creating meat, so that you don't have to harvest cows, right? But you just grow the meat in a lab, and it's exactly like any other beef. Another application of synthetic biology. Living materials, living materials is kind of this concept of you get this living cells to self-assemble into different structures. It could be like a circuit board, like say, instead of having to fabricate these tiny circuit boards, cells are already on the scale of micrometers. If you can get them to self-assemble into the layout of a circuit board, wouldn't that be cool? Because they would just kind of generate themselves and lay out the patterns automatically. And so part of my work is I want to figure out, how do we design feedback into our system to get cells to be predicted, to get them to do exactly what we want them to do. So we give some external stimuli, we have the circuit that we built and we put it into a cell. And what we want is a certain dynamics. It could be that are given by that black curve. And we want the actual dynamics to follow exactly what we built it to do, right? And we can do this can we do this using some kind of feedback? Because one of the major challenge of biology, like I said, is biology has a mind of its own. So what happens is that you think that you're going to get that red curve to match that black curve, but really what you get is something like this, where the red curve is just wildly off of your predicted trajectory. Why? Because you built this circuit, you put it into a cell, and the cell wants to survive, right? Its only mission is to replicate and survive. And when you put in something foreign into the cell, it's not going to like it. It's going to feel the toxicity of, it might be like whatever you're producing your enzymatic pathway, like if it's a biofuel, the opioids, it could actually make your cell sick. You're also using a lot of resources that the cell needs for its own growth and division. So you're almost like competing with it, right? For its own resources. And so what it does is that you then start to slow or actually kill the own cell that you need to express your proteins, to express your circuit. And so it's like this kind of negative feedback in which then the dynamics don't follow anything you expect, because there's these all changes in the metabolic pathway where the cell's trying to adapt and survive. And those are very hard to predict. And so when I think about controlling cells, or thinking about how can we do feedback control to direct cellular behavior, like the airplane, get it to do what we want it to do, despite any unknown disturbances in the environment. And so we want to think about how do we build that feedback at different levels. One level being, how do we design circuits inside the cell so that we can be robust to disturbances? How do we design at the multicellular level? And how do we design at the extracellular level? So can we give it a brain, like put a computer interface so that the computer kind of monitors what the cells are doing, processes it, kind of like our brain, right? And then decides how to change the environment of the cell in order to redirect it. So at the single cell level, just a quick overview of some of the work I've done. I've thought about what are design principles for building genetic circuits, and one of the things we looked at was the single gene oscillator. So we built this. It was like, this is one gene called LAC-I, and what it does is, so remember I was talking about like genes repressing other genes. So this LAC-I protein, you know, is expressed here. It's this protein with this blue and pink, and it can bind to that promoter site where it says PLAC over on the left. And what it does is that when it binds there, it keeps this RNA polymerase from binding, which means that it can't transcribe the protein anymore. And so that keeps it from being able to. So when it produces itself, it makes itself, it like shuts itself off, right? And so because it shuts itself off, it stops producing itself, but then it starts to like decay, there's less and less proteins, and then there's no longer enough to shut it off, so it starts to create it again. And so you get these oscillations, and I'll show a video in a sec. So, but here's some time frames where you can see a cell is like, it's glowing, and then you can see it shuts off because the proteins are dying off. And because the proteins are gone, it can start to produce it again. So you can see it glows again, then it shuts itself off, and then it glows again. So it's like kind of this oscillatory behavior. And what we're trying to explore is how changing the copy number of the gene. So this is one plasmid that holds the gene that shuts itself off, and this is another plasmid on the right, and that holds the fluorescent reporter, the glowing. The reason we can see this image, right? It's a we call it reporter. And so we're just like, well, if we change the copy number of the repressor plasmid, can we get better dynamics, better behaviors? It's kind of a analysis on the component design. And so at first we had like somewhat like 15 copies of the gene, and it looked really nasty. Like it doesn't even look like it's oscillating, right? It just looks like a total mess and a lot of noise. And then we reduced to 2 to 3 copies, and you can see it gets a little bit better. And then we reduced to a single copy, so it's just like one copy of the gene on the genome of the cell. And we got these like really pretty oscillations. So I had a video of that one. Can you guys see it? So it's like it started with like a couple cells and then they're growing, but as they're growing you can see that because each cell is inheriting the gene of the mother cell, right? They all start to kind of behave, they all behave the same and they oscillate together. But because there's no cell-to-cell communication, their oscillations start to desynchronize. And then at the multicellular level, I did some work analyzing pattern formations. So thinking about how do you design dynamics within one cell by itself, and then when you can add cell-to-cell communication so that they share information with their neighbor, how can you get these patterns to emerge that you see on the right here? Right? And so there's this like really famous paper that was written that was written by Alan Turing, and it's called Chemical Basic Morphogenesis, and it was like the first time that somebody came up with a model that created this really pretty patterns and pictures. And people were very excited because they're like, whoa, that looks like things we see in nature, right? So it was like, well, you know, the right pictures are generated just from a mathematical model, and on the left, they're pictures of actual organisms, right? In this case, fish eyes over there. And so people became very, very interested. And if we can build a model, then maybe we can understand how nature does it, or you know how these patterns arise in nature. And so I wanted to study I wanted to study different ways to get those types of patterns that we see in nature. So we have the stable localized dynamics and they're destabilized by diffusion. And so these patterns, they come from these different, they call them modes. So if you think of this as space on the x-axis, or this space on the x-axis, and on the y-axis is like, say, the color intensity, or protein concentration, or whatever state you want it to be. The first mode is like, the first mode is where it's high, and then low, and then high, low. So like how many kind of ups and downs can you get within that space? So they call them the wavelengths of the pattern, right? And so with Turing, you get these like kind of range of frequencies here, that are unstable and the rest are stable, and that's how you get these really pretty pictures. And so I was looking at these toggle switch where the one I told you about earlier where A represses B and B represses A, they put in the mouse. And I wanted to see that if we put that in a two-dimensional space, can we also get patterns that look like these pretty turn patterns? And so we first studied a one-dimensional system, and we're like, okay, well, what if we bias the left side to be high in the U protein, and then bias the right side to be high in the V? What happens if we initiate that system that way? And what happens is like this wave propagates from the left, and another wave propagates from the right. You can imagine like the left being like V is high, and then the right being like U is high. And then what happens in the middle is that you have this competition where you're like basically one side is trying to, like a tug of war, one side is trying to nudge their friend to turn blue with them, and the other side is trying to nudge their friend to turn yellow with them. So they're each trying to fight their own side, right? And so you get this a pattern where you have half one color and half the other color, right? But if you keep simulating, what is the simulation? So half is like high U and half is high V, but if you keep simulating, what happens is that eventually somebody's going to win. And so you see this line here that's in between breaks, and then they're all the same color because somebody has to win the fight, the tug of war, right? But these patterns, I won't go too much into, sorry, I'm running out of time. I won't go too much into the math, but so we did this in a two-dimensional space, and we found that this concept, this idea of this tug of war, of pushing and pulling, and me trying to get my friends next to me to be like me, creates these really pretty patterns that look a lot like the Turing patterns that were published a long time ago, but it's a very different, mathematically it's a very different mechanism that leads to these patterns, that a mechanism that you can build in a laboratory. And so that's a published work. And so we created I designed this network of like, okay, if I were to build it in the lab, how would I build it in the lab? This is kind of like the design, and actually somebody has built it already, and we did some simulations on that. And I see that you got some high frequency patterns, but then those bleed into each other because you start kind of convincing your friends to go your way, and then you get these bigger globs that persist. And then finally, I just want to talk about the last project that we're doing. It's this extracellular controller. And like I said, so we've talked about design at the single cell. How do you design circuits inside the cell? And then how do you think about designing circuits so that when you add cell-to-cell communication, you can get certain behavior? And those were the patterns that I showed. And then we'll talk about like, okay, well, how can we add a computer in the external loop in order to further enhance our control over the cellular response and behavior? And so the idea is like, by nature, cells respond to their environment, right? That's all they're doing is they're sensing and responding to the environment. And so if we can understand that mapping of how they respond to certain conditions, then we can control cellular behavior by controlling the environment that they're in, just by making sure that the environmental conditions are such that they go the way we want them to go, right? And so this is a project that I just started with a couple collaborators. So this is my first funded project. Marco Rolandi, he's at UC Santa Cruz, and Michael Levin, he's at Tufts University. And what we're trying to do is we're trying to control stem cell fate. So we have this an array of stem cells, and as you know, stem cells they can differentiate into any type of cell type. Their fate hasn't been determined yet, right? So they can, in our project, we're focusing on either creating a fat cell, a tissue cell, or a bone cell. Because what we're pitching to DARPA is the government agency that's funding us, is that, okay, if we can control the differentiation of the stem cell in space and time, then we could potentially like regrow an arm, right? like regrow the bone and the skin and the fat if you've been amputated or something like that. So these are kind of like the long-term applications that we think of. And so really what we need is that we want a certain formation. So you can see there, it's like the bone is in the center on the top left. And then you have the fat around it, right? And then the skin. And that's what we want. So what we do is we inject these cocktail of ions that will change the cell membrane, and this determines whether what cell type it becomes, right? But it may not, but what happens is that it may not work the way we want it to work. We may think that we know what we're doing, and that we think that the cell is going to do this, but it might do something completely different, right? And so that where I come in and where control theory comes in is this central brain here, where it's like, no, we need to learn. Obviously, our concept of what we thought was the input to output mapping is wrong. We need to adjust what we're introducing to the environment in order to get the right response to the cell. And so it's this entire setup that takes, a team of, right now we're a team of like 7, 8 people, where my collaborator, Marco, his whole thing is building this device, which is the actuator. The cells are grown on top of it that injects the ions onto the cell. And then the other collaborator, his whole team is engineering the cells that we're working with to make sure that they can put those fluorescent reporters so that we can see what's going on with the cell. And then my part of the project is creating the whole feedback circuit where we are imaging the cell, and I'm taking that information and I'm processing it through a control algorithm. And then my control algorithm has like some machine learning components where it's learning the response of the cell. And then what it does, it sends a signal to this actuator, and then it will say like, pump so many ions in the top left and pump XYZ ions in the bottom right portion, right? So it's kind of spatial temporal control. And so I said like we use machine learning to kind of capture the dynamics of the part of the system that we don't know how to model, we don't know how it works. And so the culmination of kind of like up to date of what I've been, my most recent research and what we've been working on in Santa Cruz. And so, yeah, if you feel, this is like what I've done. Remember I showed you all those, like how many years I was an undergrad, how many years I was a grad, how many years I was a postdoc. This work is like over the last 5, 6, like 7 or 8 years. So it's definitely a lot. But it took a long time to get a story to where you're like, this is my research, this is my field of research, this is my expertise, this is the thing that people come to me for answers, right? Like my thing is controlling biology and building feedback into these systems. So my new fun activities and struggles, the newest thing is like learning to teach. I was given a class of 200 students and nobody gave me a lick of advice. This is my second year teaching and I expect that it will go a lot better. I have been proactive in seeking out mentors and trying to gain new skills in teaching. Another big thing about being a faculty is getting money. So that project that I just shared at the end, you wouldn't believe it, but that was like $1 million. That's $1 million grant that we got. And you'd be surprised how far it doesn't go. We basically got 3 postdocs and some travel money with $1 million. The university digs in their hand, gets a good like 40%, around how much percent. And then we get the rest, and then summer salary and all these things. So you need a lot of money to keep these research projects going, to keep the labs running, and to pay graduate students. And advising graduate students through their research and personal journey. So I've been through it, and now it's kind of weird because I'm still I'm a young faculty, so I'm not so I don't have a lot of years under my belt in this mentoring game, but I also can relate more than the older faculty, right? Because I've just been there, I've been through it's very recent in my memory, the pain of going through graduate school, the pain of doing postdocs, the pain of all these things. So it's easy for me to empathize more with students and understand that things, sometimes take a little bit longer than you expect, and you're not always going to get it right away. And finding your path. So this was my personal journey, right? And not necessarily mean that that's the right journey for all of you. But you need to be strategic about your path, and really that's being really honest with yourself about what your strengths and your weaknesses are, and honing in on those. I honestly, if I would have liked to do, be a pure physicist, doing string theory, whatever, but it wasn't my strength. So I did something else, right? I was engineering, I felt like I was a little bit more natural at it, so I went with it. To be honest, my dream job probably would have been to be an artist or a theater major, but that also would not have been strategic for me. I was not that great at it either, and it probably would have been hard for me to get a job with it. So never say no to an opportunity and fear of failure. I cannot tell you how, I cannot tell you how many times I always thought I was going to fail. And to this point in my career, I still anticipate failing all the time, but I don't let it stop me. It's just kind of become a part of my life, I guess, a part of my way of being, and I just don't let it get to me. For me, it's just okay, well, if I don't get tenure, or, I don't continue this faculty job, I just go find another job, right? What's the worst case scenario is not that bad. There's no harm in trying, as long as you don't, there's nothing wrong with, okay, so let me just say this. Since probably you're not going to fail, because I've always been afraid of failing, and I have never failed to this day. So I failed here and there, that fellowship I didn't get, that school I didn't get into, little things like that. But in general, most of the things, in the broader scope of the career that I have, been working towards, I've been successful, right? And stay true to who you are and where you come from. Especially if you're trying to pave the way for other minority students and other people with similar backgrounds, you don't want to lessen the impact of your success for those behind you, right? By not staying true to yourself. So don't, just remember that. I know I had this one girl who was making a comment that she was she was African American. She was what should I do with my hair? Because she was worried that it made her look unprofessional. And this was the day she was posing this question to a panel, but I was well, and they were well, how would you mentor your student? Would you tell them to straighten their hair? And she's no. I'm no, because you don't want to propagate that mindset, right? And the whole point of you building that pathway is so that you change the culture. And to change the culture of the university, you change the mindset of this, I change the mindset of this group of people that you tend to be a minority group in, right? And turn it into a majority group. And just have the courage to make decisions are right for you. I know going to college for me was I had to go against my parents' wishes when I went to Berkeley because they really wanted me to stay in Riverside with them and didn't like me moving away. So it was a really hard decision to make, and I felt really bad for a long time, very guilty for a long time. But I know that if I would have stayed in Riverside, I would not be where I am today, and I say that with 100% certainty. So just sometimes and they'll see the benefit in the long run. Definitely, my parents are not still holding a grudge and mad at me, right? We have very good relationships and they're very happy with me. So they'll get over it. It's fine. Graduate school is a valid career option. It's not a selfish decision. It's just a longer investment. And you don't have to take out student loans anymore. And you do get paid. It's not a lot, but you get paid. I got about $30,000 a year. And I think it's a fun option to think about. And that's it. I'll do that. my first step into academia? Or what do you mean? Like, yeah. I kind of fell into it. I wasn't like, I can't say like, oh, as a kid, I always loved science. I loved playing soccer and I loved being outside.
All right.
How come you're moving on?
I think I just got into engineering because it made
Oh, sorry. I need your help.
And then science because it just became an interesting subject. I just kind of fell into it, like really my passion is so much the work itself.
Saturday, 11:30
Being the faculty, being mentoring, and being in the academic culture. I like it.
I'll teach you how to play.
I mean, I guess. Yeah, like I've been good enough for engineering. I've always like, but not good enough to be like to do like abstract geometry. Yeah, I mean like, but the level of math that we did in high school was like that was something. I do. Oh. They do that. They engineer RT. So there's like a, yeah, they're actually working on there where the engineers also attack it. Yeah, they're all targeted. They know there's one that's doing target cells and one they do with viruses, where they engineer the viruses to attack cancer. They respond to signals, so like the cancer cells have a certain marker that healthy cells don't have. And you engineer a cell to recognize that marker. It could be a protein on the membrane, it could be like metabolite that they're producing or signal that they're giving off. No, I mean, what you're designing is at the genetic level. But then when that cell grows and divides, I mean, the offspring inherits the same gene. Yeah, you're changing the DNA. And then once it starts dividing, everything has that same DNA. It's all perfect. Oh, for that part, no. Well, the stem cell, we're not engineering them. Well, we're engineering this part to put recorders so that we can track what's activated or not activated. But in that one, we're not engineering the stem cells. What we're doing is we're trying to find the right triggers to get to respond in certain ways. So, because it responds to it's response to, so it's kind of like a, you know, like if you get pregnant, you start with their stem cells, right? Like fetal stem cells. But there's some kind of signaling molecule that starts to tell it, okay, turn into a heart, turn into a liver, turn into this, right? There's an external signal that's telling you what to do. And we're basically turning ourselves into these or we tell what to do. So we're like kind of stimulating the body. Oh yeah, they've been floating for a long time. Have you ever heard of Dolly?
undergrad, did you know that you wanted to be a grad student? Because right now, I'm kind of in a window where it's like, I'm a sophomore right now. A lot of my friends are like, oh, you're undergrad, you think it doesn't matter. But at the same time, I'm not sure if I should